Okay, so finally, okay, everything is set. So thanks, Martina, for the patience. And so Martina, she's a biologist, and she's doing a postdoc at the MIT Physics of Living System group. Okay, so she's a biologist working with physicists, and she combined experiments on microbial ecology that she performed actually, and data analysis, and also modeling, because with her collaborators they develop models to describe the data. And I think it's very nice to see how also the study of microbial ecology, and also she will talk in particular about plankton and marine microbial ecology, can be studied today thanks to the massive amount of data that is available. So this is an example of how data are transforming also science. So I thank Martina again for the patience, and I leave her the floor. Thank you for being here.
Thank you, Dr. I think it's best if I stay
here. Yeah, whatever you want. Yeah.
No, just because I don't know if they can hear me. Okay, cool. So thank you for the introduction, and thank you for having me here. Yeah, so as Samir just told you, I'm a biologist in a group of physicists, so I feel quite comfortable here. It's my natural environment. But yeah, I have a background in ecology. I studied in Pisa, where I did my PhD with Lisandro Benedetti, and then for my postdoc I moved to MIT, and I started doing laboratory experiments once I started there. And yeah, so today I'm going to tell you something about my work. I mostly deal with metabolic ecology of microbial communities. And today I will show you how we can go from simple experiments to big data and back and learn important insights on how microbial communities assemble. So why do we study microbes? Well, basically because life on Earth depends on them. They make our food, they contribute to our health, and they help us fight climate change by sequestering carbon dioxide. Importantly, in this sense, there is a huge societal and scientific push to harness microbes to control or improve these functions. But in this sense, there is an important thing that we need to take into account, that all these important functions are not usually performed by a single microbe, but usually they're performed by complex communities where thousands of species interact. And so an important thing that we still need to understand is how these complex communities assemble, and specifically how the environment in which they assemble controls the diversity and function of these communities. So the problem that we have here to try to answer this question stands in the inherent complexity of microbial communities. And different. Why it's not working anymore? Okay, sorry. Now it works. Sorry, again, tons of technical difficulties. So I was saying that one problem that we have stands in the inherent complexity of microbial communities. And so different fields that are studying microbial ecology tackle this problem in different ways. For example, the people working in ecological systems biology approach the question of how communities assemble, studying the most simplest communities possible. So those formed by 2 or 3 species, and they can understand how their dynamics. And one perk of this bottom-up approach is actually that you can get a very good mechanistic understanding of how how species interact. However, it's unclear whether the simple experiments with one or two species tell us something meaningful for complex and multi-species communities like those that we find in natural systems. On the other side of the spectrum, we have the field of microbial research that instead takes a top-down approach. So basically, they go around and sample communities from their natural habitats. Then they can use sequencing techniques from 16S amplicon sequencing and metagenomics, and we can get an understanding of how of the composition of these communities. And this is the wealth of data that Samir was talking about. We have tons of data about what's happening.
I don't know if you share the vaccine or something. Share.
Okay.
No, share. Yeah, that's the one. Share. No. Top left.
Top left.
Top left.
Okay.
Okay. Okay.
Okay.
No.
No.
Stop sharing all
If I do, if I swap displays.
No. Stop share. Mi swap pure. No, vogliamo. No, non è quello che non vedo, non si vedrà. Non so, PowerPoint, poi no. Ok, però
Ma di solito così funziona. Stranissimo. No, aspetta, fammi provare un'altra roba. Vorresti vedere tutto lo schermo?
Così si vede proprio le cose. Ok, prova a fare il swap.
Non lo vedo più lo swap, il problema.
Come fai a fare?
Vabbè,
Swap e display.
sì, ma non lo vedo da qui.
Ah, vuoi che ti dico io? Dove sei?
È sul mio schermo. Oh mamma. Aspetta, magari c'è. Ora?
Sì, si vede.
Ci ho fatto? Oh. Ok.
Yes, right. Thanks. Sì,
Posso anche spostare questo qui, così.
ok, thanks.
Ora, I don't know where I was, but yeah, whatever. Anyway, there are two possible ways you can approach the problem of studying how communities assemble. One is a top-down approach. You study very simple communities with 2 or 3 species. You get a very good mechanistic understanding of what's going on, how the species interact. But the problem is that you're not able to render the complexity of microbial communities. On the other side, we have the field of microbial research. They take a top-down approach. They go around in the field, they sample communities from different people, for example, and using 16S amplicon sequencing or metagenomics, they can get an idea of the composition of the communities that they have sampled. And while here, this approach is very good at rendering the complexity of microbial communities. It's very difficult to establish causal relationships because you can sample species and communities along environmental gradients, but it's still a correlation and not a causation. So my way to deal with this problem is actually try to do a mix and match of these approaches and take what's good about them and try to understand something about how the environment changes microbial communities. So one way okay. One way to deal with it, one possible way to do this is actually to use these simple experiments with one or two species and use them to generate hypotheses that then can be tested with this huge amount of data that is publicly available. In this way, you have an idea on actually what to test using these data sets that are huge and sometimes are very difficult to deal with. A second way to, another way in which I try to approach this problem is kind of the reverse, is taking communities from their natural habitats, bringing them to the lab, and growing them in very controlled conditions. So I can change the nutrients in which the community is growing, I can change the temperature, the oxygen availability, and then I can still use the power of metagenomic techniques and amplicon sequences to measure the diversity and structure of microbial communities and link changes in the species composition to changes in the environment that I tightly control. So using these two approaches, in my work, I try to understand how the environment filters somehow biotic interactions. So how the environment sets how species interact through metabolites and other ways the species interact. And I try to understand how these biotic interactions affect the diversity and structure of microbial communities for now, and later possibly I will go into function. So about today, I will tell you, if I have time, two stories that actually match these two ways in which I tackle the problem of linking environment and changes in microbial communities. One is about the effect of temperature on the structure of marine bacterial communities, and the other one is about how the number of available resources impacts community diversity. And if you have questions, don't wait till the end. Please interrupt me. And if you don't understand, just ask me right away. Okay. So the first, we'll start with the first story about temperature. This is a project I've done in collaboration with Claire Abreu. She's a postdoc in the lab of Dimitri Petrov in Stanford. But before we were colleagues in just lab. And we also collaborated with some oceanographers, Karina Bunse and Yaron Pignassi, and that also provided us with some of the data that you will see. So why are we interested in marine communities, and what's the problem with understanding what's the effect of temperature? So we know very well that marine microbial communities change along spatial and temporal gradients. For example, in this plot, I'm showing you the distribution of the two most abundant cyanobacteria in the oceans, Prochlorococcus and Synechococcus. Alone, these two species contribute about half of the primary production of the planet. So those species are extremely important. And as you can see, they display a very clear pattern in how they are distributed in the oceans. For example, Prochlorococcus is more abundant around the tropics and the equator, and then its abundance declines going towards more temperate regions. While Synechococcus is less abundant, where Prochlorococcus dominates, but his biogeographical area extends into temperate regions. Other famous patterns that we see in the oceans are changes with seasons. And here I'm showing you an example. This is a time series of 3 years of the bacterial communities in the Baltic Sea. And you can see that, for example, these brown guys here are usually more abundant in spring, and then their abundance decreases if we go towards summer and autumn. And at the same time, if, for example, these gray guys, which are actinobacteria, become more abundant in summer and autumn. All these patterns that are very well known in oceanography have been explained using a wealth of different processes. So our question here was, is there a common principle that can explain both, for example, the distribution, the latitudinal distribution of these two species and changes in the composition of bacterial communities across seasons? And so to try to understand this, one powerful way is to use a trait-based approach. And an important trait that is usually not very considered when we think about the composition of communities is maximum growth rate. But maximum growth rate is extremely important because it gives an information about which species can survive in which environment, but also how species compete within a specific environment. And finally, also it's important because in the especially in the ocean, bacteria are classified based on how fast they grow and what kind of resource they prefer in oligotrophs, which are slow growers and thrive in low nutrient conditions, compared to copiotrophs that instead thrive in highly abundant resources and are fast growers. If you're not aware of these terms, it's not important. The important thing to remember is that maximum growth rate, it's a powerful trait to describe bacterial species. Another important thing about maximum growth rate is that it can be inferred from genomic data. So in 2016, this paper appeared in Nature Microbiology, and it's showing that the maximum growth rate is proportional to the number of copies of the 16S operon. Basically, you can go into the genome of a species, count how many copies of this operon you have, and you can then say that a species that has just one copy of the RNA gene operon is a relatively slow grower compared to a species that has 3 copies of the RNA operon. Because here we're talking about ribosomes. The more ribosomes you have, the faster you can grow. And importantly, there is a huge database that you can consult and where you can find for each species or also at different taxonomic levels, you can find the RNA, the number of copies of the RNA gene operon. And with that, you can basically extract a number that is the mean copy number that you can calculate for a community. That is basically a weighted average of the copy number that is telling you if a community is more skewed towards more abundant slow growers or more abundant fast growers. So it's a way to simplify these bars that we see in papers with a lot of species that are very difficult to interpret with just a number that is telling you who is more abundant, fast growers or slow growers. Yes.
To me, it's clear how you measure 16S or the number, but it's less clear because you have sequences in other days you can check for the number of copies. But if you have many species, I think maximum growth rate is something that's logical, so you need to measure it in a calculable species. When on the other hand, the economic data typically have own, but majority are not calculable. So I was wondering how reliable is the information on the maximum growth rate for microbial communities.
So this is a good question, of course. It's true, to get the number of copies, you need culturable bacteria. So, and many species are not culturable. The thing you can do is that it's kind of conserved. So you can pick, if you don't find species, you don't find sequences, you can go to the next taxonomic level. Of course, the far away you go from the species level, the less accurate the measurement is. We have tried, the way we do this, we match the sequence, and then if we don't find the exact sequence of the exact species, we go to the next level of the taxonomy. we tried excluding, for example, when you match at the phylum or the order level, which is the more spores, and it doesn't change that much if you just keep taxa that you can assign at the genus or family level. The only thing is that you lose a lot of data. So we tried that. Doesn't change that much our prediction, so we said, okay, it's fine. But it is true that.
So cultural check, the information about the maximum growth rate from cultural organism is a reliable proxy for culture.
This is the assumption. The other thing that we did was finding an alternative method to associate growth rate, which is using the codon usage bias, which is again inferred from the genome. And there are again measurements, but all these measurements have been done with marine bacteria, so it was a bit better for us. And the trends are kind of similar. The only problem is that codon usage bias is very bad at capturing slow growers.
I don't think you have even a monoclone.
No, indeed, it's like this, and then for slow growers, it doesn't work. Yes, exactly. And we use their methods and their database actually. It works. We still see similar trends, but especially for slow growers, it does a very bad job.
Right. The question, what is the x-axis of the
These are just different communities. So this is a drawing with this is a bar of a community. Yes, it's yes.
Define the maximum growth rate. Because it means optimal environmental conditions.
Yeah, so this is the best you can do. Of course, the realized growth rate is a completely different thing, but this is a kind of trait-based approach. You try to understand something, but the powerful thing here is that we had a prediction about what to expect that came from laboratory experiments. All right, so if this is clear, you will see this mean copy number a lot, so please remember what it means. I can stop working for unknown reasons. Okay, so basically in this project, what we did was we asked what predictions about the effect of temperature on biotic interaction we get from simple models and laboratory experiments, and then our predictions in agreement with field data. So first, let's have a look at what we know about models that incorporate temperatures and growth rate. Let's say that we have two species. One is a slow grower, and here you see the classic Lotka-Volterra competition model in which the per capita growth rate is a function of a self-inhibition term and the competition with the other species. With the other species that would be a fast grower. And then we have an added mortality term. And here we have the equation for the fast grower, basically the same thing. What we are introducing here is a dependence of the growth rate with temperature. And we use the simplest model possible, which is Arrhenius. So again, this is the classical Arrhenius form. This is the growth rate. This is the activation energy. Temperature is in Kelvin, and this is the Boltzmann constant. The other thing that you see here is basically the copy number that is multiplying the Arrhenius model. So basically, it's telling us this is how fast this species can grow. And we have the same equation for the fast grower, but in this case, the R is bigger. Okay? If you try to see what happens if these two species compete by changing in different regimes of temperature, you will see that both growth rates are going to increase with temperature, but the advantage that is conferred by the mortality to the fast grower basically diminishes as temperature increases, and therefore, as you increase temperature, the slow grower is going to be favored. Usually here I have weird looks.
Yeah. Why is there an advantage?
So this is the mortality. So let's say you have a fixed mortality term that is there and basically simulates, let's say, a dilution rate. And you see that here, this is basically the advantage that the fast grower has over the slow grower. And if you increase temperature, proportionally, the slow grower is becoming much better compared to the fast grower. And so if the fast grower was winning here is less likely that he's going to win.
It needs to be clear. What is not clear is why, like the better which is comparing this factor.
Okay, let's have a look at this. So basically, if you have this is not in this paper, it's in a different paper that was published before by my colleague. And so basically, when you go from, so here you are in the regime where the slow grower is winning. If you increase the death rate, you are favoring the fast grower, right? Because the faster you grow, the better you escape mortality.
Okay.
But if you change temperature, basically you're going in the opposite direction. And you can see analytically this by absorbing the growth rate in the alphas, and then you will see that basically the only thing that matters is the growth rate, and basically it's the growth rate. And if growth rate depends on temperature, then the slow grower is going to win if you increase temperature. But here I usually lose people because it's very counterintuitive, and you need to think about it a bit because there is mortality, which is something that is not always included in local terra model. But for example, for the ocean, you can first assume that mortality can be a global phenomenon. So there is currents that steer you away. There is grazing that is kind of agnostic of which species there are. There is viral disease, so cells are going to burst. And so we assume that all these processes can be approximated by a global mortality term.
Sorry. Can you go back to the slide? But what you were drawing, the 2 lines and the red line, it's just the arranged formula with the peak.
Yeah. The activation energy is the same.
Sorry?
The activation energy is the same for both species.
So that is only a function of P.
Yeah.
P and RS. That is what.
Mm-hmm. Yes.
Yes, but why do you assume a P? I understand the growth rate, but the effect, there should be also an effect of the interaction between those.
No, that's the thing. There is a way to to write things just as a function. So having the alphas just as a function of growth rate and death. And if you absorb the mortality in the alpha.
But this assumes that you have the same capacity.
Yes.
Growth.
Yes.
Otherwise, you cannot.
Absolutely.
But in this case, then it means that.
But we tried also changing, not having the carrying capacity fixed with the generalized local terra model, and things don't really change. So it is true this you have the carrying capacities that are bounded. Yeah.
So my question is more related to the importance of the terms that you are using, because it might be that you are writing down terms that actually are not important, like for example, interaction or the carrying capacity. So I don't understand. I mean, I understand the explanation, but I don't understand how effective is the model when you apply the data.
Okay, for the data, we have experiments showing that this is actually bad. That's what happens. What happens? That's why so this model was built to understand what was going on in experiments with 2 species.
And why do you assume that is painful?
Because in this case, we were simulating the dilutions of an experiment. When you do daily dilutions in experiments, this is the global mortality rate, but it's not crazy to assume that in the ocean you have a global mortality. Because as I was saying before, you have washing out of, of species, then grazing can be agnostic of the species. So if you have a shrimp or something actually smaller, a ciliate or something that is eating bacteria, what is actually exactly eating might be not super important. So for the ocean, a well-mixed environment, because then I will show you data of free living bacteria, it's not completely crazy. But it's true that mortality rate could be a function of temperature, could be species specific. These are all things that actually can be true. This is the simplest thing we can do, especially if you're trying to understand what's going on in the evolution experiments. Indeed, this was actually this simple model with 2 species was published before to try to explain the results of the evolution experiments. And here you can see, here we have 2 species. The red one is the fast grower, and you see that its growth rate is increasing with temperature. The blue one is the slow grower and is not really managing to catch up with the fast grower, even if increased temperature. But then when you compete them in the same environment, you see that at the lowest temperature, the fast grower is winning. Then you cross an area of coexistence. So here the 2 species are coexisting, and here the slow grower is winning. I should say that on the y-axis, we have the fraction of the slow grower. So here it goes to 0, and here it goes to 1. And this is not true just for one pair of species, but Claire and Simon tried many species pairs, and here the color code goes from the red guy winning, so the fast grower winning, towards the blue guy winning, which is slow grower. So basically, we see that in the lead evolution experiments, the slower grower is usually favored when increased temperature. And what we did next was trying to see, okay, what if instead we have hundreds of species? So we basically, Claire did run simulation with using a generalized form of the lockable data model with competition with our usual mortality rate. And here we still have the growth rate dependent on temperature with this R term that indicates the copy number. And the distribution of copy numbers of these simulated 100 species comes from a geometric distribution where the parameter P represents the fraction of species that have copy number 1. So it actually has a biological meaning. And we see that usually the fraction of, there is a quite high number of species that have copy number 1. And if we run these simulations, you can see that the mean copy number.
What about that? Alpha?
The interactions, no, they have a, I don't remember what is the average, but it's a matrix with the average probably was 0.5 and 0.5. The distribution, it's random, yeah. Yeah, yes. I can give you more details on the parameters if you want, and also they are all written in the manuscript. Yeah, if you run this, but we tried the changing alpha, so we tried extending the parameter range and see where this prediction is actually verified, or we see this negative trend. But so it's quite robust, and we see that the mean copy number decreases with temperature. And again, we can calculate this mean copy number because for each communities we have the abundance of the relative abundance of the species and the copy number at the end of the simulations when the communities are stabilized.
So at time 0, we draw the copy number from the distribution, and this is for parameters equal to this plot.
0.8 something.
Okay, if it's a P,
yes.
So they are, you run the simulation, you see
when they are stabilized. So they run for 800 hours, something like this. So they are quite long. It takes a while to stabilize. When they're stabilized, we calculate the mean copy number, and this is what you see if you run for different temperatures.
How many species?
100.
Yeah, but you could have extinction, right?
Yeah, we have extinctions.
So what's your
Here the diversity, we don't really have any, so we know we don't have a prediction for diversity, or at least not yet. But here the only thing we are looking at is the structure, so the relative abundance and the copy number of the stabilized communities.
You have a label of the copy number attached to the species, which
Yes.
is abundance, and then you come to the
Exactly.
that downward slope would still remain if the slower growers would not outcompete the faster growers at high temperatures, right? Because at low temperatures, the slower growers would go extinct, but if they survive at high temperatures,
No, not all of them. So you still have, you have less slow growers compared to fast growers.
okay, now, but asking, is the presence of slow growers will break down the copy number,
Yes,
even if they don't outcompete the fast growers?
true. And actually, this is a good point for data, because when we to actually look at that, in the data, the real data, not the simulations, we excluded all the copy number 1 from the data, the marine data, and the trend is still there. So even if you remove these tons of species that have, actually those are the most abundant in some cases, you still see the same trends. But it's a good point. We didn't do it with simulations, we did it with the real data. Yeah, so given this, we have actually a nice prediction to test in field data, and that the proportion of slower growing taxa, and so the copy number should be smaller during boreal summer, at the equator and the tropics, and at the surface of the ocean. So to try to see whether this was true, we started collecting data sets of marine communities. And this is just a map showing 3 of them. We have a total of 7. And so this is the 8 years time series in the Baltic Sea that was provided by our collaborators that are in the paper. Then we have this amazing transect that crosses the Atlantic Oceans and spans basically 100 degrees of latitude. And also, here the communities are sampled in the first 200 meters of the water column. And then we have Tara Ocean, that is this cruise that is going around the globe and collects samples, not just at different latitudes, but also at different depths. So the samples are collected between the surface and 1,000 meters. And we use each of these data sets to try to see whether our predictions are verified. So now I will show you in each of these plots how temperature varies. For example, here in the LMO time series, in LMO data, it's a time series of how temperature varies with time and how the mean copy number varies with time. And we see you see, of course, that temperature is higher in summer and it's lower in winter. And somehow also the copy number mirrors the variation in temperature, but on the opposite. So you have the highest copy number with the lowest temperature. And we can do the same thing for the Atlantic transect, the ANT, and we have, of course, temperature is higher around the equator and the tropics, and nicely also the mean copy number basically mirrors this distribution. And finally, this is a portion of the Tara Ocean dataset that basically is a chunk between 0 and 40 degrees of latitude that takes all the measurements across the water column. So here is the temperature going from, this is 25 towards 14, 15 at 1,000 meters. And here is the copy number mirroring the temperature, how it goes. So, but if you're not convinced yet, you can plot the mean copy number as a function of temperature. And these are the 3 data sets, and these are the slopes. And you see these are all negative slopes. And importantly, the slope is kind of the same, which is kind of, to me, it was really, really surprising that the only thing that is changing is the intercept. And you can imagine that the intercept is actually related to the amount of slow growers that are in these communities. And just to say a thing that to me is quite relevant, the Baltic Sea is the coldest one and has also the higher intercept. What we did next was trying to use the model to fit the data. And what we did was basically only fitting the parameter P, that is the only parameter that you need for the geometric distribution describing where we fish the RNA copy numbers. And here you can see the colorful dots are the actual data, and the black line is the fit that we obtained from the model. And you can see that we do a pretty good job at basically getting the data with just this super simple model with just one parameter.
P is fixed once for all the or for all the arguments for all the.
Yes, it's one P for each data set. And this is we start with the initial P, and this is the final distribution, which is okay. We have, it's not perfect, of course, but given that it's super simple, I think we can be pretty satisfied. So the black is again the fitted distribution, and the colorful one is the real distribution of the copy numbers with their abundance. And I would say that the worst we do with the AMT, but the others are pretty good. And also we are decent in predicting the slope. So just with the simple model and the P, we can recapitulate the slope that we actually get from the real data.
Sorry, when you say simple,
The l'ortogoterra. And the only parameter that we fit is P.
so the interaction, I mean, what depends on
There's no dependence on the interactions. The only thing that you need to know is P. That's the good thing.
so basically you can use any media.
I don't think so, but no, okay. Actually, yes. So in principle, you don't need the interaction to see this.
Okay, in this case, so they behave neutral.
I mean, neutrally, you don't expect an environmental forcing to drive what you're seeing, right?
No, neutrally in the sense that each species is a kind of independent of the other.
Okay.
I mean, if what you say is what happens here, I think that once we come up with a neutral moment with the parameter depend on the parameter of the temperature, we get more or less the stuff.
Well, I think that would be fine as well, because we're just saying that we have the strong effect of temperature that is driving, that is how we understand the composition of the species. This is the, yeah.
In other terms, let's say, does not really matter how you fix the distribution from which you take the alpha.
I mean, there are,
Of course, if
yes.
you take values that have been extinct.
Yeah. And also the mortality rate, it's actually quite important. If you go to crazy mortality rates, you don't see this. So there are regions of the parameter space in which this doesn't work.
But let's say once you're in this region, you can even in the limit of putting the alpha to 0, probably the delta, which
This is an effect of the temperature, not of the community.
Yes, of course. Yes.
Everything.
Is the temperature on the growth rates?
Yes. So the community by per se is not important.
I mean, if you mean the identity of the species or the interactions, I mean, the interactions that are not really important. The identity, yes, because it depends.
No. Of course, the identity is because of the trait.
Yeah. Yes, that's what we're trying to say. And the other thing that, the other thing that you can say as a critique to this is that it's not just temperature that is changing across these latitudinal and seasonal gradients. For example, this is the environmental data for the Baltic Sea. You see the temperature has this nice trend, but also this is the dissolved organic carbon changes, the other nutrients, nitrates, And so you can probably hypothesize, well, maybe the effect of temperature is spurious and it depends on other environmental variables. So what we did was the statistical analysis using generalized additive models, in which we asked, okay, if we add the effect of the all the other variables, is the coefficient of temperature still negative and statistically significant? And this is true. The only thing, unfortunately, for the Atlantic Transept, they collected data, but they were not available, so we couldn't really add other variables. But for LMO, we have a ton of variables, and also for Tara, we have phosphate and oxygen.
Do they correlate with that?
Sorry?
Do the nutrients correlate with them?
They actually do the opposite. But so I actually bet, okay. Sorry. What I mean is that they are anti-correlated, and they are anti-correlated. So but you can ask if you are adding things that are correlated, whether this is kind of worsening the performance of the model. And I try to do that, and you have, if you keep the nutrients inside, the model is still good. So there is a correlation, but it's not affecting the performance of the model. So you can check this with the variance inflation factor. So there are things you can do. And the other thing is that we tried it also for other data sets. For example, this is the data set, it's a time series in the Pacific Ocean, and this is a time series in the Mediterranean, which is the only one that is actually a bit shaky in terms of our predictions, because actually they have this variable is called day length, so it's the number of hours of light, which is actually strongly correlated with temperature. Because in summer you have longer days, and this seems to be the most effective, the most important variable. But it's also a data set that has a lot of slow growers, and I decided not to dive into this here. But as I was saying before, if you eliminate from these data sets all the species that have copy number 1, just to verify that we're not just tracking the distribution, the biogeography of slow growers, all these trends are still negative and actually become more negative. Meaning that for actually relatively fast growers, these trends are actually better. And this is, to me, due to the fact that we are very bad at estimating the growth rate of slow growers, because there is a huge chunk of species, especially the Scorsai level clade, that they all have copy number 1, but they actually live in different places of the ocean. They have there are different ecotypes, but since we cannot capture them, we are not able to understand the differences in their growth rate, as I was saying before. So there is a problem. You want to add something? Which were no. Okay, just to conclude this part. This principle that we are drawing is in agreement with the distribution of Prochlorococcus and Synechococcus. So what we're saying that the slow grower is favored by increasing temperatures. Prochlorococcus is a slow grower compared to Synechococcus. So their distribution is still consistent with our predictions. And finally, interestingly, there are warming experiments of soil communities, and most of the times what they see is that the mean copy number decreases. And usually they explain this by saying that temperature reduces the availability of nutrients, but actually there is a simpler explanation, is that temperature is actually favoring slow growers, and this is possibly a reason why you see more slow growers after you warm soil for 20 years. So to conclude, I hope that I convinced that we discovered a macroecological pattern in the distribution of fast and slow growers in marine communities, that this pattern was somehow surprisingly predicted by a simple generalized local model. But to me, one of the most important things is that we are actually providing a prediction for how marine communities can change with warming. So we can expect that with warming oceans, we can see larger proportions of slow growers in marine communities. So if we still have time and I have not killed you, we can probably go on, switch gears, and okay, I can do it. Okay. The second story is about another important environmental forcing for the structure and diversity of microbial communities that are available resources. And this is a project that is already published, and yeah, we published it last year. Okay, so why trying to understand how resources affect community diversity? Mostly because we have a lot of predictions from ecological theory, and they usually clash with the results of the experiments. So, for example, the principle of competitive exclusion says that if you provide one limiting resource, then there will be only one species surviving, even though there are several ways we can avoid this happening. But actually, when we do experiments and we provide a single source of carbon, for example, glucose, what we see is that there are multiple species coexisting very happily. And one possible explanation for this is that if we are in a well-mixed environment, there is no special structure and so on and so forth. These species, by consuming the supplied resource, leave in the environment other metabolic byproducts that can be then used by other species. And so multiple species can actually coexist. And this phenomenon is called cross-feeding. But there are still a lot of open questions. For example, experiments have been tried with glucose, some amino acids, and but not with a huge variety of carbon sources. Another open question is what happens if we increase the number of resources? What's the relationship between diversity and the number of resources? And so what we in this project, the question that we were trying to explain is if there is variability in the number of surviving species in different resources, how can we explain this? And also, what is actually the relationship between community richness and the number of resources that is supplied in the media? And to respond to these questions, I performed experiments with the second approach that I told at the beginning, by taking communities from a soil sample that I basically went downstairs from the building at MIT. I scooped a bit of soil and I came back to the lab. And in the lab, you shake the bacteria off the grain of soils mechanically, and you obtain a microbial suspension. And later, I actually sequenced that. And at the beginning, there were about 750 species. And then I used this microbial suspension to inoculate different resource environments. And I had 75 combinations that derived from the combining different resources and different number of resources from this pool of 16 carbon sources. And you can recognize some simple sugars. These are the saccharides. These are organic acids, sugar alcohols. This is a kind of an amino acid, but it's produced a lot by roots of plants. And finally, these are huge polymers, cellulose and starch. And all of these resources could either provide it as a single source for carbon or in combination of 2, 4, 8, 15. And then I had the condition in which I threw all the resources together. But importantly, the concentration of carbon across all these conditions was the same. So there was no change of carbon. And then to grow these microbial communities, I followed the daily dilution protocol, by which you let them grow for 24 hours, and the next day you fish a bit of this culture and inoculate it in a fresh batch of media. And I did this for 7 days. And we know that usually this is the right amount of time to have communities that are stabilized. And at this point, I did 16 sample sequencing, and I measured the diversity of microbial communities and their structure. So now what we will see for the next few slides is the plot in which you have the richness of the communities on the y-axis, and on the x-axis, the number of supplied resources. And here I added the prediction that would come from the competitive exclusion principle. So the x equals y line. So this is the upper bound to what the competitive exclusion principle predicts. And now I'm adding the richness of the communities grows in single sources of carbon. And here each color represents the average of 3 replicates for each carbon source. And you can see that there is an average of 20 species, which is already saying this doesn't work. But also there is a huge variability in the richness of these communities. For example, there are these 3 communities that support just a dozen of species, while, for example, cellulose and sorbitol can support up to 40 species. And since in this environment we have basically no special structure, we are stirring them, so there is enough oxygen. One possible way to explain this, the coexistence of multiple species is breastfeeding. And also the other thing that we were expecting, since we saw so many species in single resources, that diversity would skyrocket really fast in the moment we add more resources. But now I'm adding the richness of the communities with 2 resources, and diversity didn't really increase. Actually, kind of diversity did not significantly increase. And so now I'm adding also the results of the other combinations. And you see that the community diversity increased slowly, but with a strong linear relationship. So basically, increases 1 by 1 with the number of supplied resources. So at a constant rate. And the thing that was very puzzling is that you have basically the same number of species in 15 and 16 resources that you have in single resources. So what I'm telling you in the next following slides is trying to understand what causes the variability in richness of the single resources. So, and to try to understand that, we went back to cross-feeding, and we said, okay, what if we can try to understand how many metabolites can be produced but if we have 1 resource in the environment? And here you see this is a map of the generic metabolism of a generic ensemble of species. And of course, it's simplified; it's much more complex. But you can recognize the glycolysis. And then this is the lower glycolysis and the TCA cycle. And you can see that there are some resources that enter in this part of the map, while others enter on this other part of the map at the level of the TCA cycle. And what we asked was, okay, if I have this genetic ensemble of microbes and I have, for example, and I provide, for example, sucrose, how many metabolic intermediates can be produced by this generic ensemble of species? And to answer this question, we use the scope expansion analysis, which basically yielded this plot. So this is the number of predicted number of metabolites that you can produce starting from each of the resources, and this is the richness. And we saw a very nice correlation, which is basically telling us that the metabolic network of that is triggered by the supplied resource explain how many species can coexist in each resource, and basically helped us understand why we see this variability in the diversity of communities. And the second question that we asked is whether, okay, is this information helping us understanding why we see this linear relationship with the number of available resources? And the answer is yes. So what we did was using a consumer resource model, Pankaj Mehta style. So the consumer resource model with cross-feeding, but instead of using a random cross-feeding matrix, we use this map to inform the cross-feeding matrix. And the assumption that we made is that each species consumes preferably one resource and leaves in the environment the two byproducts that are adjacent to the consumed resource in this map. The map looks more like a network, but this is just a drawing to understand what's going on. These two intermediates will be consumed by other species that will leak other intermediates and so on and so forth. And using this metabolic network to build the cross-feeding matrix, we were able to basically recapitulate the linear relationship between the number of supplied resources and the richness of the communities. Another thing is that I want to add is that you can look at how species are distributed across communities. And we were able to identify species that could be found in all the combination of resources, and we call them generalists, and other species that instead the specialists were present only in few resources. And in this plot, I'm just showing you the richness of the communities divided in these three groups of species: the generalists, intermediates, and specialists. And importantly, the model is also able to kind of recapitulate that. So, and what it's telling us is that let's say the core number of generalists is what sets the intercept of the relationship, while the specialists are the species that are actually contributing to the increase in diversity. And this was quite fast, hopefully. And so with this project, we were able to say that the identity and number of supplied resources actually determines the metabolic network and community diversity. And also that a consumer resource model is able to recapitulate our results, experimental results, but we needed to incorporate the metabolic network. So, and this is telling us that metabolic interactions are extremely important to understand how communities assemble. And finally, what is interesting to me is that in principle, these results support the idea that we can modulate the diversity of communities by changing the number and type of resources in which they grow. And with this, I'm about to conclude. Basically, I've shown you how temperature and resources can set biotic interactions. There are other important things that we can look into. I've just looked into carbon sources, but nitrogen and phosphorus are also important. Another important thing to check is the availability of oxygen, because it sets which metabolisms are available in the community. And so there is more to understand about the diversity in structure and function. And also, I think there is a way to basically build an ecological toolbox for microbiome manipulation, which I cannot see, but it's fine. And with this, I would like to thank my advisor at MIT, Jeff Gore. Akshit and Kian helped me with the resource project, and with input also from Jacopo Grilli, Marco Martina, and Marco Cerigliano. And for the project about temperature, it is Claire, Karina, and Yaron. And thank you for your attention.
I have a question about the story. When you show the special generalist pattern, you say that with your model you can calculate the kind of behavior. But this behavior is strictly related. What I want to say is that not the cross-feeding, but the direct interaction between species and the resources. Set, let's say, who decide the cross-feeding are specialists and generalists, right? So you can set that one species consume only 2 resources, no?
No, in the model is actually everyone is a specialist, so consumes one resource. But here is generalists are species that are found in all the communities, not just they are not associated to a specific resource. This is more a habitat occupation way of defining generalists and specialists rather than I'm consuming a lot of resources.
We assume that the one that is generalist.
Our idea is that the one that is generalist consumes something that is always produced, like the intermediates. So the
starting point anyway about from the direct let's say interaction resource species is that it's basically kind of the animal matrix where each species consumes directly only one resource.
Yes, but
among the 16.
Yes.
Then all the other is due to cross-feeding.
Not just one resource of the 16, but also one resource among the metabolites.
One resource of the metabolite.
Yeah. And the idea is that, for example, citrate and fumarate are both resources that we supply and resources that are usually produced, because once you start with glucose, you go ahead and you do the full circle. And so they are usually produced. So that's why, for example, we see these generalists, we believe that are those that eat, that consume the TCA cycle, which was informed also by the experiments, because there are the pseudomonads. If you look into the structure, they are known to consume organic acids. They were dominant in the organic acids, but they're always present. And our idea is that somehow they are best at exploiting these resources, and since these resources can be usually there, that's why they are always present.
How do you know that the 16 total carbon and what they're only in the soil?
No, this is no, that's not.
So 16 are what you supply.
Yes.
But then why do you expect that the exclusion principle to work? Because the total number of resources is this entire the nodes in this metabolic network, right? Not the 16.
No, can you say it again? I don't think.
How do you count resources? You count resources on the type of metabolite, or you count resources?
No, I was
Exclusion principle. These are 16, no, these are the 16.
No, but what are they taking? Yeah, but why would they not also be counted?
No, they are counted. That's why you see more species.
Right.
So the idea is that you can basically infer from the number of metabolites how many species survive. And this works very well on a almost one-to-one relationship in single resources. But when you go into the multiple resources, this doesn't work well anymore. Otherwise, what we would see, so the number of metabolites do not increase linearly with the number of resources, but actually they increase they grow fast and they reach a plateau later. But the thing is, with this type of models, you assume that every time you consume something, you make a fraction of it. So down the chain, there are metabolites that are present at such a low concentration that cannot be consumed. And as you increase the number of resources, but you cap the concentration of carbon, more and more of these metabolites are not available to be consumed. And that's why you see this linear relationship. So you in principle have more metabolites than species that then survive.
But can you measure the total number of people metabolites in the community, or at least or
It would be very nice.
Okay.
No, I asked. That was the original idea, to go and check how many metabolites with the metabolomics. But if you do an untargeted ones, you get a lot of peaks and you don't understand. Basically, I asked them, they told me it's totally a disaster. And
curiosity about experiments, since you cultivated many communities, I guess You did it by hand or also kind of automation?
We have,
Carry on.
it is my favorite machine. So we have this, it's a liquid handler that, so instead of using one pipette, you have 96 tips.
Mhm.
And so you go from, for example, you have to do the daily dilutions, you can transfer 96
Okay.
from, but all the media with the resources is by hand. So you wait and then
So
you,
is the time where you provide one of the 16
yeah, like the media you have to do it by hand,
Okay.
3 days.
Thank you.
No, but it's worth. I
Yeah.
guess.
Okay, so thank you again. I think you have anything else. Thank you.
Thank you very much. Bye.